Good morning, good afternoon, good evening, and welcome to Live from Technion, webinars with Technion experts. My name is Shanta Mali, and I'm the Chief Marketing Officer at the American Technion Society. Whether you are joining us for the first time or the 8th time, this marks our 8th webinar. If you're from California, New York, Israel, or Australia, thank you for being here and for your interest in the Technion. As you know, the Technion's faculty and alumni are leading Israel in the world in research and innovations aimed at solving global challenges. And what better time than now to have such talented and dedicated scientists? The current COVID-19 pandemic poses an immediate and unprecedented threat, unlike anything we've seen in our lifetimes. And lucky for us, the Technion is on it. For today's program, Closing the Loop, Technion COVID-19 efforts from diagnostics to therapeutics, we are proud to have two Technion superstars addressing these urgent needs. Associate Professor Moran Bercovici is a member of both the Technion's Faculty of Mechanical Engineering and Faculty of Biomedical Engineering and is head of the Technion Microfluidic Technologies Laboratory. He earned his bachelor's degree in aerospace engineering from the Technion at the age of 18 and completed his master's degree at Technion during his military service. Professor Berkovici won a Fulbright doctoral fellowship to Stanford. He conducted postdoctoral studies at Stanford Medical School and returned to his Technion roots in 2010. Associate Professor Josué Sznitman wears many hats at the Technion. He is the Associate Chair for Undergraduate Studies. He heads the Technion Biofluids Laboratory in the Faculty of Biomedical Engineering and directs the Norman Seiden Multidisciplinary Graduate Program in the Russell Berry Nanotechnology Institute. Professor Sznitman earned his bachelor degree from MIT and his master's and doctoral degrees from the Swiss Federal Institute of Technology. In 2010, Professor Sznitman joined the Technion as the Horev Fellow in Leaders in Science and Technology program funded by the Henry and Marilyn Taub Foundation. 2010 was a good year for the Technion. Both Moran and Josué are leading priority COVID-19 related projects. Professor Berkovici's team is focusing on testing. Professor Sznitman's team is developing therapeutics. I'd like to take a moment now to express our gratitude to several ATS supporters who have made specific gifts to the professors' projects. We're pleased to recognize Alberto Hamwi, Gil Zamer, the Lou Duntz Trust, and Victoria and Rob Pollock, who have supported the research of Professor Sznitman. Thank you. In addition, we'd like to thank our donors who are supporting the work of Professor Berkovici's team and his research. Moving into the webinar, if you have any questions for the professors, please use the Q&A feature at the bottom of the window. All questions will be held until the end of both presentations. Please note that today's webinar presentation will be recorded. Professors, we're thrilled to have both of you. Thank you so much for taking time away from your families and work. And with that, I will say, Professor Berkovici, please take it away.
Thanks, Shanta. Thanks a lot for this kind introduction. And hello, everyone on all time zones. Thanks for joining us. This is an unusual webinar, an unusual time, but maybe this electronic format is a good way of really bringing a lot of people together. I wish I could see your faces and responses and read the audience a little bit better. So feel free to use the chat and the Q&A. I'll try to glance on these also in real time and get a bit of a feel for the questions that you have. And of course, we'll have more time for questions at the end. So before we get going, Chantal already did a great job at introducing me, but I want to give you a little bit more background where I come from, and also this gives context to the work that we're doing. So I'm originally Israeli, born and raised in Haifa, literally 10 minutes away from the Technion, which was lucky for me. After getting kicked out of high school, Technion opened its doors for me, and I'm 38 now, but I'm already since the age of 15 essentially at the Technion. So yeah, 23 years at the Technion in this way or another. I did my undergrad in aerospace engineering. My passion for the field of fluid mechanics. So this is the control and motion of fluids. This can be liquids or gases. That passion really evolved during my years in aerospace. And then as part of my military service, I served as an engineer at Rafael. Rafael is a defense company associated with the Israeli military, working on projects that have to do with fluid mechanics. And in the context of defense, these are things that fly, occasionally explode. I think you can get the idea. And that's in aerodynamics, right? So it's the flow of air around bodies that makes them fly, have lift, minimize drag, etc. During my military service, I served for 5 years. I also did my master's, which was also still in aerospace engineering, but I started going to things that are at small scales. So my research work there was trying to have create very small disturbances, small, the size of about 100 microns, the thickness of a typical hair, on the apex of different aircrafts, and using very, very small perturbations to the flow, be able to induce large vortices over the aircraft, and in this way, essentially maneuver them without having any large surfaces. So this took me to the micro world. The last thing I did at Rafael before leaving for a PhD at Stanford, is now allowed. We're allowed to mention that, and that's Iron. What's today is known as Iron Dome, and those were very exciting times to contribute directly to defense and protecting people in Israel, and I'm very proud of that time in my life as well. But I switched gears to a PhD in the field of microfluidics, so going to small scale, so that's fluid mechanics, but on scales that go from hundreds of microns, so like I said, that's the width of a hair, and those are the large scales, and down to the scales of hundreds of nanometers, so 1,000th of that scale. On those scales, fluids behave in a very different way than they behave in what we know from everyday life. There are new forces that are important there, especially electrical forces. And we started going into that world, which is fascinating from a physics point of view, but also from the applications that it can bring. During my time at Stanford, there was still is a very nice project called BioX, where bio stands for bio and X stands for anything else. And that project really aims to take engineers and physicists and introduce them into life and medical sciences. And I got really fascinated by that field. Just the challenges, the opportunities that exist in life and medical sciences were mind-blowing to me. And so when I finished my PhD, I was looking to educate myself a little bit in that field. I've never taken any biology or chemistry class in my life. Someone was kind enough to host me in the medical school, Professor Joe Liao. And if you look at in medicine for fluid mechanics, which again is my passion and expertise, then basically you can find hematology, right, associated with the flow of blood, or urology associated with urine. And I found myself in the urology department, spent there quite some time, basically taking engineering concepts and trying to apply them towards medical diagnostics, which essentially brought me to what I'm doing today, the way I define myself today, is really leveraging engineering principle approaches to solve problems in the life and medical sciences. So after that time, I came back to the Technion, Department of Mechanical Engineering, and started my lab that I call the Microfluidic Technologies Laboratory. So my lab in academia, we have a prerogative, which is, we're not developing a product. We are studying what are sometimes very fundamental problems, like I told you, the basic physics of how fluids behave at small scales. And that's an important scientific contribution. It has merit by itself. But then as an engineer, I can't help but think, okay, and now that we understood this fascinating new phenomena, what can we do with it? And in a way, we throw the arrow and we draw the target around it when it comes to applications. But when we do that, things that are often much more different or more creative come along than directly looking to solve a problem. And so I'm telling you this story because really the solutions, the things we're trying to do now for diagnostics of the coronavirus started from very basic, physics, understanding, investigations of what happens when we apply electric fields to special chemistries at the microscale. There are all sorts of interesting shock waves that occur there, and we studied them publishing in physics journals and applied mathematics journals. But then we saw that when we throw molecules into that setup, we can actually focus the molecules very significantly, meaning we can increase their concentration by hundreds, thousands folds, and create very rapid reactions between molecules, accelerate reactions by 10,000 folds. So that's taking a reaction that normally would take a couple of days and reduce it to a few tens of seconds. And that's all based on the basic physics. And I say that really to emphasize that they're two sides of the same coin, and both are very, very valuable. So my lab has been working on diagnostics for about 6 years since I joined the Technion originally. We developed several assays that are all based on this ability to take a very small sample. The focus is on very, very small samples, extract molecules of interest from them, and molecules of interest being, for example, nucleic acids. These can be DNA or RNA, proteins, and focusing them, increasing the concentration, and allowing in this way a very rapid detection. One way to think of that, of why it allows very rapid reaction, every biological reaction, biosensing, is based on a probe, a molecule that we design, that is designed to attach to some other molecule of interest and give us a signal. Now, when the concentration of the molecule that we're looking for is very high, then imagine this room, the Starship Enterprise here, is your vial where the experiment is happening. If you have a lot of molecules in the room, they're going to meet each other very quickly and give you a signal. That's what happens at very high concentrations. But as concentrations go down, the chance of your target molecule meeting the probe goes down, and that's what makes the detection become very slow. By taking our microchannels, so we're looking at chips that are basically, you can hold them in your hand, between your fingers. It has microchannels that are a few hundreds microns in size. You put in the sample, you apply the electric field, the molecules are collected, focused, and using that, we essentially have speed dating of the molecules, because they're all confined in such a small space that they all move around, confined to that little space, meet each other very quickly, and give us that signal. During these first 6 years, we were working primarily on developing these new methods for diagnostics, and this is one of the things I've been taught well at Stanford, that writing patents would not guarantee that anyone will use your technology, but not writing patents absolutely guarantees that no one will ever use your technology. And so we've been writing a lot of patents on these technologies. We've actually 2.5 years ago, we started a company around this. The technology was licensed. Unfortunately, that company closed. And around the same time, we started doing. We got a large grant from the European Union and started doing other things that have to do with single cell analysis, which I'll tell you next time we meet. But now when the Corona crisis started, it was like an aha moment. It's everything we've worked on so far is exactly right for tackling this problem, because for many diagnostic issues, there's the question, okay, so you can make it faster and you can make it more sensitive, but large labs are pretty good. And now suddenly, all of a sudden, there is this very clear need that what we need is very rapid diagnostics that could be distributed and done everywhere, and not necessarily rely on those centralized lab. The standard method in which the detection is done today, the labs, you all read about it, I'm sure, all the time in newspapers, is using PCR. It's a marvelous technique that takes the virus RNA and creates additional copies, which can then be detected very easily. But the process of taking the sample, preparing the sample, meaning cleaning it up, before it can go into that marvelous process of amplification, is a multi-step, very tedious process that takes the time. In addition to the fact that it has to be done in a central lab that adds all the bookkeeping, the samples that get lost. At least in Israel, we have this issue of it takes days until a person gets the result back, and so time suddenly became very critical. And so our focus in the lab is really on making the fastest assay possible. Our target is on the 5 to 10 minute time scale, which is what I think is the time scale necessary if you want to bring the economy back to something that is semi-normal, meaning you go to the airport and on check-in, you do your swab, and before you get to the gate, you have the result. You don't board the airplane if you're positive, obviously. You go into a football game, you go into an important meeting, you get tested, you get the result on the spot. So timescales of hours are irrelevant there. We want and we can get to minutes. So when this started, we basically dropped everything. I recruited back some of my old team, people who also graduated, but were eager, to now use everything they developed toward this. I have to say that it's an international effort with a lot of openness from people really all over the world. The scientific community is open. Information is flowing. Supply chain is difficult, especially in Israel. We're an island. But we are collaborating on this also with IBM Research in Zurich, which while the clean room at the Technion was closed, we were still running. Josué's lab was running, a handful of other labs. The clean room was closed. We were getting fabrication from IBM Research in Zurich because they understood it's important. friends at Stanford were designing probes for us. Really an amazing international effort. And so I won't bore you with the technical details, but I'll just tell you that we're testing two main approaches for this rapid diagnostics. One is our method for direct detection of the viral RNA. It is the same target that the PCR is currently using, except we don't go through any of the cleanup, any of the amplification. We, by focusing, are able to pull out the RNA molecules from the sample and detect them directly. And just about a week ago, we got finally everything together, the chips, the specialized probes, everything together, and we can actually do RNA detection in the lab. Our next step is going to clinical tests on this with Rambam Hospital. That's the hospital in Haifa that is also most strongly affiliated with the Technion. One of the challenges is actually going to be now, it's good news, but the number of patients in Israel is declining very rapidly. We're very happy about that. It's going to make testing more challenging, but we'll find other places, maybe in collaboration with the US, to do additional testing. The other approach we're trying has never been tried before. It's a very new patent that we just submitted a couple of weeks back. And that is detecting directly the virus itself. So the virus is 100 nanometers in size, which sounds small. It is small, but is much bigger than other molecules, such as antibodies that can label it. And we developed a method that if you take probes, these are your antibodies that attach themselves to the virus, all you need to do is mix them, throw them into our chip, apply an electric field. The chip is configured in a way that it simply pulls out just things of that size, 100 nanometers, pulls them out while leaving all the rest of the gunk back in the reservoir. And so we think that that is potentially the most minimal approach for doing the detection without any sample preparation. There are big questions around this, however, because no one in the world is targeting that as a diagnostic. Everyone else is doing essentially the RNA detection. So we don't exactly know how many intact viruses are in the samples, etc. So there are questions beyond the technology. It goes back to the clinical samples and whether it's useful once we apply, but there's no way of trying other than moving forward. And so I'm looking at the time now and realizing that, as always, professors love to profess, and so I probably went over too long. I'll just end with saying that I'm excited to have the opportunity to try and contribute to this. I probably won't save the world in 2 weeks. These are not the timescales for these events. But I would hate to find out that in the fall we're experiencing another wave, and I would then regret not starting earlier. So as soon as this started, we dropped everything and we're on it. And I'm, like I started saying, really privileged to have an amazing lab with the infrastructure, equipment, the excellent people, the excellent collaborators, like my friend Josué, I'll introduce in a second, where anytime we need anything, we're good friends, above everything, and things flow very quickly. Very privileged to be in that situation, to have the opportunity to try and help and tackle this problem. With that, I see there are a few questions, but I'll transition and hand the microphone virtually to my very good friend and colleague at that order, Professor Josué Sznitman, who is an amazing scientist, and I learned so much from him over the years, both on the science, on his approach to things. And I have to say, above all, he's a real mensch, and it's a privilege to be on this seminar with him. Josué, Stage is yours.
That I'm blushing? Can you tell? This is the last time you call me Professor Moran. I insist. It's true that Moran and I go way back. I will be celebrating my 10th year this July since I made Aliyah. Unlike Moran, I was not born and raised a few streets away from the Technion. I'm born in France. I was born a French citizen. I moved that as a child to the United States, and around the age of 10 or so, my family relocated in Switzerland, in Zurich. So I have, as of today, something like 3 nationalities. My children have a 4th one. They happen to be Norwegian. I won't get into the details. I'm often asked how I got to the Technion and how is it that I got into the field of research relating to lungs. So I'll try to answer the first one. People ask me, what did I do here in Israel? How did I get here? And I tell them it's my kind of one and only running joke, which is I came for the job. And it's actually quite true. It's not even a joke. I came to Israel around the time, so in 2010, but that happens to be after the economic crisis was hitting hard in the United States where we were living at the time. And I was trying to make way home, my way back home to Switzerland. But as many of us know in academia and in many other professions, you don't pick and choose always where you want to be. So I had to extend a little bit my horizon and came across a job offer as a professor at the Technion. And little did I know that this would be the start of my next step in life. And here I am, a decade later in Israel with first generation born boys who are 8 or 9 years old. They'll be 9. And I cannot emphasize how unpredictable life is. I heard on the BBC the other day something that struck a deep chord for me, which was, it came from Hinduism, I suppose. It said something like, in life, you don't really know what you get, but it's how you react to it. And in many ways, COVID-19 is a bit of climax in my research because just a few months ago, literally, I was touring California for the ATS, and I was announcing publicly the work that was relating to ARDS, acute respiratory distress syndrome, which seemed to kind of spike a lot of interest because of this concept, which I'll elaborate upon, which is this foam therapy. Little did I know, little did any of us know, that within a few months, this would suddenly be at the forefront of our research efforts. And I'll get into that. So about the second question, which is, why on earth am I doing lung research? I will say that in many ways, research into lung therapies and lung disease is often seen as a niche topic. And that has to do, of course, because whatever we believe might be the most interesting thing on earth, it certainly isn't, and I'm perfectly fine with that. But I think that it's often overshadowed by other medical issues, cardiovascular, neuroscience, and so on and so forth. And I've been very comfortable with that. It's okay. But I actually have a very similar trajectory in the core of it with Moran. So it happens that you've invited 2 scientists who share a common background, which is again that fluid mechanics. So I hope you've made it thus far in terms of understanding what it's about. But Moran came from the side of aerodynamics in aircraft, and I just happened to be in love with aerodynamics of race cars. And the reason is simply because I find race cars really just cool to look at. But their shape answers the laws of aerodynamics. The thing is, my dream of making that car company didn't exactly pan out. I didn't marry the daughter of Ferrari, but it's okay. But that curiosity in aerodynamics led me to seek a direction where I thought there would be more direct benefit on a kind of human level. And so I sought pragmatically a direction of research where I could merge my curiosity and interest in this topic of fluid mechanics and, again, aerodynamics with something to do with the body. So Moran mentioned to you urology. So I was a little bit less passionate about urine, I must say. But I didn't go for the world of cardiovascular flows. And that has to do with a very pragmatic choice, which was for me, I was coming from a group, a research lab that had absolutely nothing to do with medicine. And I just called a shot and thought to myself, I have no chance of doing anything in a world as dense and as ripe with big names as in the world of cardiovascular flows, in other words, blood flows. So I sought my direction on my own in the world of respiratory flows, which is breathing. And that's my history, that's my story. This is why I'm here. So we talk about therapies, and I'm going to say just a few words about what on earth we're doing and why is it relevant. I'm an engineer, like Moran. I'm a mechanical engineer. I'm not a pharmacologist. And so the principal work that I am involved with is leveraging engineering and bioengineering techniques that do involve, just like Moran does, things that are biological, but I do not develop myself therapeutics. I try to leverage existing therapeutics or therapeutics to be that come from other places and find neat ways or original novel ways to deliver them. So how is this all combined with this COVID-19? Unfortunately, by and large, the people who ultimately will die from COVID-19, at least at the moment, are the ones that fall in the category of something like acute respiratory distress syndrome. In practice, these are the patients that we talk about that have moved on to ventilators. Now, you need to understand that when it's time to be put on a ventilator, things are not going well at all. It's in fact a symptom, or in other words, trying to tackle a life-saving approach, which has to do, a machine is basically going to do the job for you. So you can imagine that by that stage, things are not at all well. So not unlike other viruses, the coronavirus, SARS-CoV-2, happens to attack our respiratory system in many ways which we still poorly understand, but a consequence of which is that you start having potentially, and we don't know why some people do and others don't, but for those who do have serious respiratory issues, it's a terrible syndrome that is the consequence of a number of factors, which again, we do not fully understand. But it's kind of like elements of a pneumonia that go so bad that at some point you have so much inflammation of all sorts with an immune system that is completely whacked and is infiltrating from everywhere your lungs, and the cells of your lung start to malfunction to a point that basically you don't even have the right material inside the lung to help support your own muscles and let you breathe. And then by then, it's time to get yourself on a ventilator. Now, we understand that with the crisis that's just happened on the planet, there was a tremendous lack of ventilators, which is also telling you that in general, we do not need as many ventilators under normal circumstances. Now, the awful truth is that when it's time to be put on a ventilator, and in particular, those who need to be basically anesthetized, and in other words, they are not conscious anymore and are on these ventilators, it's basically a race against the clock. And what it means is that we're trying to buy time because we do not have a therapy available. And this, what we're witnessing in COVID-19, is something that is analogous to acute respiratory distress syndrome, which is a syndrome that arises when either a pneumonia, for instance, goes terribly wrong, worsens, in other words, or you've had an accident as a car accident, and it's such a terrible shock that basically it busts your lungs, and so you have whole organ failure, or drowning, or inhaling fumes from a fire, and so on and so forth. It is not a disease, it is a syndrome. But unfortunately, in ARDS, just like in COVID-19, we don't have therapies to the very day. So where do we enter in this? One of the hallmarks of conditions such as ARDS is, as I mentioned, many of the lung functions do not work. And one of the things that are very important in the lungs, which is the essence of how we're all here, is that our lungs are not dry. There's a little liquid in them that lines it, which is called surfactant. And it's not unlike soap. It is certainly not soap, but it shares one common property with soap, which is the reason why we use soap as a detergent, is because it has an easiness to infiltrate all the places in your clothes, in our dishwasher, in the dishes, etc., to get rid of dirt. And even in an environment that's not wet, which is an environment with air. And this alleviation that happens thanks to surfactant, which is the kind of the property of the soap part, because surfactant is a very complicated and complex biological fluid that the lung cells produce. But the bottom line is, among all the problems that we can face in ARDS and COVID-19 induced ARDS, or alike, an analogous syndrome, is that surfactant breakdown means that our muscles cannot exert enough force to distend and relax the lungs. Now, ARDS has a sister syndrome, and it's called IRDS, which is infant respiratory distress syndrome. Because, ladies and gentlemen, babies that are born prematurely, their cells are not yet mature, and so they cannot secrete either at all or sufficiently this so-called surfactant. So babies cannot breathe on their own because their muscles are not strong enough to compete against an environment that needs to be distended but does not have that soapy material on them. Okay, but the thing is that in IRDS, although it's an invasive approach, we have a therapy that works 98% or so of the time for 30 odd years or so. And that's to inject surfactant inside the lungs of babies, of these prematures. Now, the prematures, and I'm not a big man, but a premature is probably the size of my fist here. So the lungs are probably the size of my nail. Now, when you instill surfactant inside a lung that's the size of our nails or so, or the tip of our finger, this installation, in other words, delivering this liquid through the throat, which by all means is not something pretty to watch, but it is life-saving in basically all premature babies. It works, and by some magic of biology, which none of us understand, not only does it immediately alleviate this burden of trying to breathe with our muscles, it triggers also slowly the cells to start making their own. But, and this is the big but, is for the past 30 years or so, or 50 years since we've come across ARDS or put our finger on saying what it was, we don't know how to scale this for adults. And the hypothesis that came from my group was the following. And I can't even take the credit for it. I can only take the credit for having hired people who are a lot smarter than me. And I think Moran will agree that that's probably the best gift in our job, is that satisfaction and gratitude of picking people who are far wiser and more novel and original than we are. And the hypothesis was that if liquid doesn't disperse inside lungs of adults, it may have to do with the fact that gravity sucks and it pools basically the surfactant in specific areas. And we do not witness this in a premature because the lungs are so small. But in something like a balloon the size of my chest, then trying to reach all the areas is very complicated. So we needed to think of something which is potentially surfactant works in adults, but it's never been proven to do so. But it needs to be administered in a way that gives it a chance to reach these huge adult lungs. And so came the idea of this liquid foam therapy. In other words, why not foam surfactant? But you know just as well as I do that soap likes to be foam, does it not? So that's what we went about, was to basically find techniques to foam surfactant. And by the way, surfactant therapy in general is done by administering animal surfactant that's extracted in general from cows and giving it to babies. So the idea was to foam surfactant and administer it instead of injecting it through inside the trachea in the throat in a liquid form. So now the chronology. 4 years ago, I'm giving you the quick version, is when this all started. In 2017, by then, we had put down the provisional patent for what is called LIFT, liquid foam therapy. And then the European Union, not unlike Moran, as well as the Ministry of Science and Technology of Israel, and the Polak Fund, which was the first one on board. Actually, chronologically, I think the Polak Fund was the first to believe in us. So I've had a chance to thank the family in person. The idea was to show that there's a chance for this. Now, we, again, are talking about ARDS. None of us knew that COVID-19 was going to loom. So we got money to run the first animal experiments. And these were completed, believe it or not, and this is insane, in January. Again, COVID-19 was not on our horizon. But what we had shown by then was that in rats, which is the first benchmark of kind of getting a medical and scientific community approval, was to start with a model of ARDS. So we induce ARDS in rats. So what you do is you remove, and again, it's not pretty, you do a lavage, you take out from the lungs the surfactant, and you induce the state of lung function breakdown. So lo and behold, what we showed is that if you do surfactant therapy as a foam, it is safe and it works. Of course, it works like liquid because a rat is not so much bigger or smaller than a premature baby. And so suddenly we're in February, and basically the storm hits planet Earth, and we were sped up at full speed to try and prove that this will work in humans. And what you need to understand is this is meant to be a rescue therapy for people who are on ventilators. It is not a cure to SARS-CoV-2, in other words, to COVID-19. It is to give a chance to people who are about to die, because ARDS is notorious for having something like a 40% mortality rate, okay? Because there are no therapies. You give oxygen and hope for the best, okay? Is that by giving the surfactant, you can at least buy time for the body to start tackling the virus. Because unless you can overcome your organ failure, which then leads to multiple organ failure, because if the lungs aren't working, then there's no oxygen going to the rest of the body, and there's a catastrophic storm of inflammation everywhere, which is a very complex problem, which I don't understand. But if you can't at least start by tackling the lung problem, there's little chance you'll get around the rest. So as we speak, the Vice President, Kobe Rubinstein, I don't know if he's here with us with the 233 people. What happened is that he basically took the bull by the horn and said, what do you guys need to make this go faster? And so we said, we need to now prove that if it works and it's safe in the rats, which is a very important step, we need to prove that it can work in lungs that are the size of adults. And so as we speak, we are preparing the experiments now in whole size pigs, which is about to be conducted in the weeks to come, maybe within a month, because we need to finalize the protocols in the Faculty of Medicine down at the beach in the Rappaport faculty. And so the premise, the hope, and listen guys, I don't know, we don't know if this will work. But before we can move to humans and save potentially people who are on the ventilators, we need to show that it will work in the pigs. And so in the few months now to come, we need to have a few positive results that give us some confidence that the foam surfactant may be a trick as a rescue therapy, and surfactant has a chance to be the rescue therapy for the so-called acute respiratory distress syndrome, and then get ourselves back on tackling COVID-19. Okay, so as we speak, this is happening in live. In science, for me, things don't happen in live. That's not the way our communities usually work. So we would not have announced so publicly all the results of the rats because the scientific community hasn't yet reviewed our publication because we were in the midst of writing it. So you imagine for us, it's kind of a very, very quick chain of reaction. But that's it, ladies and gents. In the best case scenario, let me put it frankly to you. In the very best case scenario, I don't think it's foreseeable to see within less than 10 months, a year. I don't think we would get an approval to run anything in humans. Although maybe, and there are some discussions, that it could be used as a compassionate treatment, which is not something very bloomy, but it is potentially something that could be a difference between life and death. So on a hopeful note, I guess this is an opportunity for people in my field, and I'm not alone, to rise and shine. It's important that I nor Moran, none of us mislead anybody. But if our hypothesis turns out to be right, there is a chance that liquid foam therapy could be a game changer in saving the lives of people who are already in the final stages of the ventilator use. More broadly, and there is hope that liquid foam therapy could revolutionize, in part, other delivery systems for lung diseases, which I'm not going to get into today, because it is extremely potent as a delivery platform that avoids bringing things through the blood. How is the foam surfactant administered in the rats or pigs? It's basically a machine that foams the surfactant, not unlike your Nespresso machine that makes your foam, but it uses, in essence, it connects onto the ventilator because everything needs to go through the trachea. So these are people who are already intubated, okay? There's no way out of it. So this is meant for intubated patients. So with that, I will stop with my story now. We're living it live in real time, and with time permitted, I'll be happy to take on some questions after, as I see there are some in the chat.
Thank you, Professor Berkovici and Professor Sznitman. We are so grateful to have you. We have a couple minutes for questions, and I will introduce Jerry Kleinman, our ATS Executive Director of Principal Gifts, who will moderate the Q&A session for us. Jerry, are you available? All right, he'll get us through as many as we can.
So thank you. I received 3 or 4 notes on my cell phone from some of the ATS donors who had helped both you gentlemen through awards and lab setups and recruitment funds, and they're so proud of you. And I won't mention all their names, but they really are astonishingly proud of having helped bring 2 scientists like yourself to the Technion, and I just wanted you to know that.
Thank you, guys.
You're
Thank you very much.
The way I read this is that we've gone from Iron Dome to urology and race cars to lung therapies. And I'm trying to weave something here, but I have a question for Josué on smoking and vaping. And maybe it's for both of you, but can the virus be carry from one person to another by ingesting vapors and smoke in society?
I think that might be better for Josué. My answer is I don't know, so maybe Josué knows.
Josué, what
Okay.
do you think?
So that's actually very much up my alley. I came from the world of aerosol therapy. So this whole world of aerosols and particles. While there's a great misunderstanding at the level of the details of how the disease is spread, one aspect that we are quite confident about, and it's a general acknowledgement from all over the globe in the scientific communities, is that coughing, breathing, etc., sneezing are definitely vectors to move things around. In what proportion, how much, how many viruses inside are the drops of basically the vapor coming out of our mouth are communicated? How far do they reach inside the lung? Is it first stuck inside the nose, at the back of our throat, and then migrates? We've been knocking our heads against the wall and trying to decipher through the literature. There's no simple answer at this stage, but let us be very clear. While there is the potential to transmit and carry the disease from surfaces, in other words, from touching and then putting to your mouth, certainly if somebody's breathing in your face within less than a meter or a feet or something, and you sit around that person, even if they don't sneeze or cough, the odds are likely that you will transmit. Yes.
Interesting. And sobering at the same time. Moran, I have a quick question, 2 questions. Well, one's a quick question. You mentioned getting kicked out of high school. Do your teachers know what became of you?
I hope not.
But you had mentioned a call to action, getting your colleagues to come back, to set up the lab, to pivot, if you will. What was involved in that under these difficult circumstances? Because that must have been happening while the Technion was shut down.
Yeah. So first of all, some people I contacted, and some people I just didn't manage to contact because they contacted me first. Like, Moran, can we? This is it. Can now I apply? What my PhD was to this. And so people were able to come back to the lab. Some people could not come physically because of kids, etc., but kept being consultants on the phone. To other team members who are in the lab, maybe never ran that assay, but now we're essentially extension of the hands of the people outside. And so we have a very active Slack, if you guys know it. It's like a WhatsApp for labs that are exchanging very quickly information and messages, etc. So we have very extended Slack of everyone, and this is how information is transferred very quickly. Now at the Technion, acted very swiftly to have, like I said, I think at the beginning it was a handful of labs and later extended beyond that, of labs that work as a response labs to Corona. Josué's lab was one, mine was another. And those got an exemption essentially from the government and were not shut down. And so I think we were down for literally a day or maybe two until people figured out what's going on. And then again, it's the same. The Technion management, Kobe Rubinstein, is also the head of research at Technion, acted to enable running the labs. And so we had everything running, a limited crew because of the regulations, but the full supply of import into Israel of things, deliveries on campus, and even cleaning services just particularly for these regions that were active. So we were rolling from day one.
Interesting. Josué, someone has asked, what is the downside of trying foam surfactant on humans now if they're already in such trouble?
So the short answer is that it's very, very rare that you get a green light to just try a vaccine on humans after no tests whatsoever before, via pilots on animals and passing on to humans. We cannot, there is no mechanism, and rightfully so, to jump the steps and go from preclinical animal experiments to clinical human experiments. I will say this, that our project is being run in parallel in China with collaborators, and it could very well be that they will be the first to leverage our technology and use this technique as a compassionate treatment. But in the meantime, there's no way around going for the large scale animal experiments, large animal experiments. It's just a no-no.
Someone has asked, because they've seen this before, the relationship to research for defense that finds its way to biomedical treatments. And they were just curious about your views on, someone had said almost so much of it starts in the defense of Israel and ends up in defense of human life, if I quoted the person correctly. I just thought to think about your experiences and your thoughts about an institution like the Technion and its role in pivoting from one to the other. Moran, do you have?
Sure. Yeah, I think it's, it's Israeli. It's the Israeli story, right? We rely on defense in Israel for our existence, and these things, they break barriers. Wars, as terrible as they are, the positive things that comes out of them is that barriers are broken, and it creates a rush for creativity and making new things. Defense is complex. It's also it's an integration of many different aspects that need to come together to really provide defense. And the medical field, as almost the opposite, right? I mean, in the medical field, we wake up in the morning trying to think of way to save people. Very different from a war. It's similar in the sense that it's such a complex system that requires integration from so many different parts, that I think that similarity really allows people to go from one to the other. The crisis with COVID does feel in many senses like many other wars we've experienced in Israel. that's, you know, everybody springs to action. We're used to improvising under these conditions and trying to come up with solutions. Where again, it's a lot of it is on personal connections. A lot of the things that I've been able, you know, to push through is through my previous life connections in the defense and people, you know, from all over. Going back to it's the Israeli story. Everybody knows everyone and pushing things this way.
So I have just a couple more. One quickly for Josué. Someone had looked up your CV and noticed MIT and the Swiss Institute and Penn and Princeton and finding your way around the diaspora. And they were curious about what surprised you most or attracted you most to the Technion.
I will say something that has to do that overlaps very nicely with what Moran said. You know, I'm here a decade, and I like to joke around. I'm kind of that eternal tourist in Israel, kind of a misfit. I'm too polite. I'm too organized. I'm not balagan enough in Israel. But
we love you, Josué.
What?
We love you.
I know. Yeah. So this man makes me blush.
Sorry.
I think the strength that I witness in Israel, and it connects deeply with what Mohan says, is, and I come from a place where, you know, in Switzerland, resources are there. There's generally little worry. Here, people do extraordinary things with limited resources. There is an appetite and a culture of bootstrapping and thinking outside the box. So I always like to look at it from two sides. I never understand why bureaucracy is so complex when it should be straightforward on many levels, which is what I know from other places. But on the other hand, when things don't fit that red tape, then things happen here precisely because people are so in tune with having to troubleshoot constantly that it makes them so original in breaking away from challenges. And this is something you don't see much in Europe. And it's a very, very deep strength of the system. And it, of course, ties in with the history and the survival, and that, you know, there's not always a silver spoon on the table. And it shows. And for somebody like myself who's built a lab here, it's incredible at the Technion, the amount of originality, novelty, and appetite and fire in the eyes that you witness, that I've had the luxury and benefit to piggyback and surf on that wave. And I think for me, this is the pinnacle of my scientific career, is having had the fortune of meeting these kind of people.
So I will turn it back to Shanta with one comment, and I'll quote somebody who just wrote in a few minutes ago. You make us very proud. So I echo that. I thank you, gentlemen, and I turn it back to Shanta.
Thank you, Jerry. Thank you, professors. I echo everything. You make us proud. I think that is a great way to conclude this session, and thank you. To our audience, just to note, if you are interested in supporting the COVID-related work of either of our professors, please message us in the Q&A message. You can do that now, and you can send an email to programs@ats.org. That's programs@ats.org. We've recorded the session. We'll be sending this to you in an email, the link. And please join us next week, where we'll speak with Professors Marcel Maklouf and Ezri Tarazi. And the title of that webinar is Betting on Out-of-the-Box Ideas to Fight COVID-19 related crisis. So thank you so much, everyone, for being with us. And again, thank you, professors.
Thank you very much.
So much.